Epigenetics is about self, it's about identity. And what I mean by that is that if you look at your body, look at your skin cells, think about your eyes and your nose, they're all different. They have an identity. And the reason they're different is because of developmental history and because of what we would call gene expression, which defines what they are. Now, as a consequence of gene expression changes, there are little chemical changes on the DNA and chromatin that's encoded in our nucleus that generates the expression patterns. Those are like little chemical tags. Now, if you think about barcodes, when you're buying food on the shelf, you see all those little black lines, and they're all unique. So the store knows where everything is, it knows its turnover, it knows everything else. Epigenetics essentially is putting a barcode in every cell in your body. It knows exactly what it is, so it's about identity. Now, why is that interesting even from biology? Because it's a real-time readout of what something is. That is a very powerful tool. If that external influence has an obvious effect that affects the cell physiology, that changes its gene expression, you're going to change the barcode. And so the possibility is that by looking at the barcodes, it's actually diagnostic of the type of change, the type of external influence that you've had. Now, what we want to know from a modern environment now, we're exposed to a lot of modern chemicals, is if we get exposure, can we measure that by looking at real-time changes in the barcode? If we take away the exposure, does the barcode reset? Is that indicative of everything being all right again, or does it persist or have a memory of it, so it may be sensitized to a second exposure? So for that reason, the having it there in the first place and being able to define it, and then seeing how it changes in response to exposure, to age, to development, to gender, to the stress of a modern 24-hour life, that again is potentially very powerful, because in our terms it may be diagnostic of how fit you are, how healthy you are. Just think about the cells of our body as being all different. The germ cells are as different from any other cells, any other cell type. Now, to go through the special processes that require you to make different types of male and female germ cells, that involves repacking your DNA and then bringing it together again, the next generation, that is associated with a lot of changes in the barcode. Now, I would argue that in those situations, the barcodes that emerge after that process are essentially wiped out and they're set up again. So in one sense, if that is the case, how can epigenetics, if you define it that way, be transgenerational? It can't, because you have, if you like, a quality control service that operates every time you reproduce, that wipes out, with the exception of specific regions, wipes out the profile so that you start with a clean sheet in each generation. Now, if you think about why would you need to do that, there's a very good evolutionary reason, because if you carry over a barcode from one generation that is associated or affects expression states, it's going to perturb the processes that you want to do for early reproduction, everything else as well, continuing. So I would argue that from an animal, a mammal, a human, a mouse, a rodent point of view, that is a very rare and very almost probably impossible event. Now, there are other species where that's different. For instance, in plants, where they do manage to transmit information between generations, if you like. That's not necessarily just encoded by DNA. But that would be talking about an entirely different situation compared to animal studies and human existence. Why it's so brilliant and so applicable to safety study is because of this idea of having a barcode of what is normal and what changes. So if you can devise assays that are sensitive enough, you can essentially work out real-time and transient changes, not just endpoint changes after a long-time exposure. What happens in that process? So that tells you more about physiology, it tells you more about cellular changes, it tells you about modes of action to the exposure that you've seen. Why does it happen in some tissues and not others? What's different about them? Are there receptors in one tissues that are responsive to this chemical that are not present in others? So it's extremely important from the point of view of the toxin industry, because it gives them a new tool. The other side of that is that many of the tests that are used at the moment take a long time. They require a lot of animals. So if we had a test that was shorter, that was more reliable and more useful in identifying biomarkers that may be based on barcodes or maybe based on something the barcodes have identified, we should be able to reduce the time for those tests and maybe perhaps improve our screening so that you would have a reduction in animal use, for example, or a better utilization and employment of animals for the tests we want to undertake, so that we don't have to do the same thing over and over again. We can do it, we can be more sure and more predictive of what's going to happen with the chemicals we want to test. You're going to get many different definitions of epigenetics, and not all of them are going to necessarily agree, because there is still argument about, what is epigenetics, how it occurs, how is that barcode perturbed? Is it because it's primarily itself changing things, or is it a response to environmental exposures or changes in cellular composition? I would definitely be in the field where I would see epigenetic changes as a response, but because it's a response, again, it's a fantastic tool for working out what's going on. It has a very, very important role in what it chose to do, because it's an interface in one sense between industry, academia and the public. Now, the pace of scientific change and scientific knowledge that's going on at the moment is phenomenal, to the extent that we are generating more data than we can easily interpret or accumulate and work out what's going on. So we're talking about cutting edge technologies telling us more about something new. So that has to be explained, that has to be perpetuated throughout society as to what that means, because essentially we are changing the paradigms for how we think things are working, how we understand things, and how these are going to be useful in terms of how it describes our well-being and how it describes our environment that we're living into in terms of chemical exposure. I can see it now, I can see how it's being so useful. So if we take the more extreme case, where if you take disease studies, now especially cancer, which has, there's many different types of cancer. Most of them undergo epigenetic change that is different from the tissue of origin from which they've arose. Now those changes are diagnostic, they agree with pathology, they agree with clinical outcomes, clinical descriptions, which is very good because we're talking about a molecular signature, which means we're all talking about the same language. So we're increasing knowledge, that's the first thing. At the same point, this jigsaw we get with these new epigenetic changes that we see in disease give us huge clues to how this has occurred and why it has occurred. What is actually the genetic condition that has allowed the cancer to propagate and occur in different tissue types. So what that does is gives us new diagnostic tools and actually allows us to identify the molecular event that takes place to develop the cancer, the molecular events that precede and go after that event, and perhaps what is the best type of therapy, what we should be targeting on if we want to actually really control this cancer or kill it. If you think about that, what that's going to mean societally and everything else, that's going to change a lot of things in terms of what sort of society we're going to live in from 50 to 100 years' time from now, if all those things come to fruition, if we're still doing that investment, if we still think that doing science is a really great idea, which obviously I do, if we want to improve our world and make it a better place to live in.